All right, good afternoon, and thanks for all of you who are not from here and came for this workshop. We really appreciate it. Since the name of the workshop comprises the electron microscopy, we thought we also should tell you something about the electron microscopy itself. However, and also, since you're all probably structural biologists and are interested in the structural biology applications, I will do something completely different, and I will try to point you to the applications you can use the electron microscopy for, which are not related to or aimed primarily for structural biology. So the advantage, one of the advantages of the electron microscopy is that it can allows you to study a sample across a large range of sizes. So we can go from individual animals, even living animals, inside the electron microscope, which are more than a millimeter in size, up to the single angstroms resolution in the modern structural biology applications. So for this part, or for my talk, I will primarily try to focus on this range, so that I'll give you some overview what you can use the electron microscope, or what all kind of the samples you can place into the electron microscope and have nothing to do with structural biology. To start with, just briefly introducing when electrons interact with the matter, they can either pass through or they can be reflected from your specimen. And therefore, we define two types of microscopies. It's either a transmission electron microscopy, which primarily uses the elastically scattered electron which pass through the specimen. And for such a microscopy, you need to have a very, very thin specimen because the electrons interact strongly with the matter. And once the sample is more, let's say, than 1 micron thick, all of the electrons will be trapped within your sample. We can also use the electrons which are reflected, either as a backscattered electrons or generated as secondary electrons by the interaction of the primary electrons with your sample, and those are the primary source of the signal in the scanning electron microscopy. So this is just a brief overview how the electron microscopes look like. In both cases you have the source of the illumination and then a condenser system which generates a beam interacting with the sample. In the transmission electron microscope, we primarily use a planar electron wave that interacts with the sample, and then the image is formed by the objective lens and then magnified on the camera or the fluorescent screen. In scanning electron microscope, we primarily generate a focused beam, a small spot, electron spot on the specimen, and the microscope is equipped with a scanning coils that allow scanning of the electron beam across the sample and image the sample. Of course, both of these types of microscopes can exist in a form where they can do both. So the TEM can do also scanning if you equip it with the scanning coils, and the SEM microscope can also work in the transmission mode if you put your detector below your sample, having in mind that your sample has to be very thin. So the general microscope is scanning transmission electron microscope. Specifically in the scanning electron microscope, there are these two types of electrons we primarily detect. These are secondary electrons generated at the very surface of your specimen, and they primarily bring information about the morphology of your sample. These electrons primarily have very, very low energy. On the other hand, the backscattered electrons can penetrate deeper into the volume of your sample, and bringing the information from deeper in the sample, and also you can use them to learn something not only about the morphology, which is part of that information contained, but also about the chemical composition of your specimen. If you're confused and this doesn't make sense, in general, the TEM is the big one, and the SEM is the small one. What's more, there is also now yet another microscope that you can include into your scanning electron microscope, and you can have the source of ions. So instead of looking at the sample or illuminating the sample with the electrons, you can, for instance, use gallium ions to image the surface of your specimen. The gallium will work the same way. Of course, it will generate the secondary electrons. It will generate also secondary ions. But thanks to its mass, which is much stronger, much higher than for the electrons, it will also more effectively remove your specimen. And this is one of the applications that is widely used in the material science electron microscopy, and recently and now is gaining a lot of attention also in life science electron microscopy. So then you have something which is what is called a FIPSEM microscope that has a electron column and also the ion column. And the ion column can be not only the gallium, but nowadays also so-called plasma sources are gaining more and more attention in life sciences. So what I'm going to talk about is what kind of samples we can image in a SEM, and I'll try to tell you something about the specifics of the sample preparation. Then decided to tell you something about block phase imaging, and of course in the end something about the cryo techniques, because that's the hype of the recent time. And as I mentioned, I will not go into any structural biology, as we have much better speaker to cover this topic afterwards. All right, so as I said earlier, the scanning electron microscope can be used to study morphology. So for instance, as in here, you can see this is the pollen of a plant, and you may be interested whether the pollen of the plant which is grown here in Czech Republic differs from the one that is grown in the Mediterranean region. And that's exactly what's the case here. These are geographically the same plant, but grown at geographically distinct regions. We can either depict the whole image, or we can get to the much higher level of detail to see individual pores. The sample preparation in this case is super simple. You just take the pollen, which contains very low residual water. You air dry it and put it on the sticky tape, place it into the microscope, sputter the surface with a thin layer of metal, 1 to 2 nanometers, and to make the surface conductive, and then you can directly image the sample. The other example shows that we can go across a wide variety of scales. So this is the plant female organ. It is 2 millimeters in size. So, and you can also depict it in the scanning electron microscope. But then you can zoom in into the level of individual cells, scaling this range of magnifications that I tried to show you earlier. For this particular experiment, since this contains more of the residual water, the imaging in this case is done at cryo conditions, but before imaging we do so-called sublimation. So the residual water is sublimed from the specimen, and the sample prep contains just a quick freezing into the liquid nitrogen, which is not the vitrification, of course, the water will freeze into various forms, including the crystalline one. But when we place it into the into the microscope, we have a stage which is cooled to the liquid nitrogen temperature, so we can keep the sample at the liquid nitrogen temperature for further. This part is a part of the phase diagram of the water. In the standard scanning or transmission electron microscope in the sample region, the pressure at the cryo conditions is around 1 to the minor power of -5 pascals. So there's a 1 to the minus of 7 in millibars. And if we have the sample at the liquid nitrogen temperature somewhere here, you see that the water itself doesn't sublime from the sample. It's rather your sample gets contaminated, and that's what you normally see. That during the time, if you leave the sample in the SEM, the water will condensate on the tap and contaminate, specifically in the microscopes with the large chambers. However, if we manage to raise the temperature above to the sublimation region, we can sublime out most of the water. Then we can sputter again the surface with the metal to increase the conductivity of the sample and improve the imaging. The last example is the imaging of the plant chromosomes. In this sample, for this sample, the sample preparation which has been done here is very similar to the block phase techniques, which will follow. The chromosomes are extracted from the plant roots, chemically fixed by glutaraldehyde and formaldehyde, contrasted by either platinum or uranyl acetate, and then the residual water is replaced for ethanol or acetone, which have the lower surface tension, and after that either the sample is air dried. However, the air drying might cause some damages in the sample. Therefore, specifically for the second scanning electron microscopy, we sometimes use critical point drying. That's where you replace the residual acetone with a liquid carbon dioxide. And the good thing about the liquid carbon dioxide is that you can reach its critical point with the standard or with fairly normal laboratory conditions. So at slightly increased pressure and at around 30 degrees, the CO2 reaches its critical point and evaporates, and that will effectively dry your sample completely. After that, you can either image again a non-sputtered or metal sputtered sample to get the information regarding the morphology of your chromosomes in this case. So in the next phase, I would like to touch the method that allows you to reconstruct a 3D volume of your specimen. So we can work with the larger volumes, and this primarily comprises embedding of your specimen into the resin, followed by the slicing and imaging in TEM. This can be done for very large samples, macroscopic samples, but it can also be performed for the cell suspension in order to learn about the structure of the cell at the ultra-structural level. The major advantage of this approach is that it is fairly sensitive, and the sample preparation protocol is fairly robust, and you will get 3D representation of your object of interest. Still, we work at the non-physiological conditions, use fairly toxic chemicals, and the level of the detail we can reach is limited by the whole procedure of the sample prep. So the sample preparation comprises again chemical cross-linking for which either primarily use either glutaraldehyde or formaldehyde in a, say, 2% solution in a water. You incubate that with your sample for a certain period of time based on the volume of your sample. And then you use additional chemicals for fixation in order to stabilize the membrane. You use primarily stabilize and contrast the membranes. You use osmium tetrahydroxide. You can further enhance the contrast of the proteins by uranyl acetate or subsequently, for instance, by lead citrate. Then the sample undergoes the dehydration. Again, one can use either ethanol or acetone series and slowly replace the water in your specimen for these low for this either of the two. The problem with the dehydration is that your sample shrinks, and you may get an extraction of lipid membranes, and that's the point where then the aggregate artifacts may appear in your sample. And finally, the acetone is replaced by the resin, again, which is again done stepwise as a mixture in general of the resin with the propylene oxide and the slow infiltration of the resin into the sample, followed by baking your sample for some time in the oven. So if you pass that, you will end up with a sample that is in such a block, and at the tip of that block you have your specimen sitting. And what you can do, you can use this instrument, which is an ultramicrotome, and slice very thin slices, which are 50 to 70 nanometers thick, let's say. The slices will come on the water level, and then you can manually pick them up with the TEM grid, which is a standard carrier for transmission electron microscopy, and transfer such a sample to transmission electron microscope for imaging. So the advantage of using this approach is that the TEM, in contrast to SEM, has in general can reach high resolution. So you can reach higher resolution. You can also do tomography, so you can reconstruct the 3D, even 3D volume of this very thin section. But still bearing in mind that your resolution is limited, because whatever you're looking at is not primarily your protein, but the signal coming from the chemicals you used for the staining, so either the osmium or uranyl acetate or the lead. So therefore, this method doesn't allow you by itself to target, for instance, individual proteins inside, easily target individual proteins inside the sample. However, once you have the slices, you can also use additional either EM contrasting agents, such like nanoparticles or fluorescence markers, to interact with the surface exposed residual proteins functional group. And then you can fairly precisely target using the correlative light and electron microscopy the regions of your interest in these slices. An alternative approach is the use of the scanning electron microscope. Here you have the chance again to use the mechanical slicing and have the ultramicrotome directly inside the microscope chamber, and cut the slice image with a scanning electron microscope, get an image, cut yet another one, etc. The alternative option is to use FIP. So you can use the You can use the gallium ions to ablate part of your specimen, generate a phase that you can then image with the electrons. In a scanning electron microscope, we primarily, nowadays, with the recent technologies, primarily use the backscattered detect primarily backscattered electrons because of the optics of the scanning electron microscope has improved, and now we have the immersion lenses which can boost the amount of the electrons that are dragged back into the electron column. The advantage, or maybe advantage, of the using FIP for slicing is that you can make much thinner slices. You can, for instance, do the 10 nanometer slicing and generate or obtain the Z information bit more precisely than with the mechanical slicing. On the other hand, removing one part of your specimen takes a certain time, and in contrast to the mechanical slicing, this approach is more limited by the size of the sample you can image. So if you would like to, if your sample is a tissue and you need to image the whole large tissue, then this approach, either in combination in TEM or SEM, is probably preferable. Whereas if you're interested in looking into the single cell, then this approach, the combination of FIP with a scanning electron microscope, might be easier. Both ways. The utilization of SEM microscope might bring advantages over the mechanical sectioning for TEM, because the process will directly give you individual slices of your 3D volume, one after each other, without the need of fishing the sections on the TEM grid, which is not error prone, and then if you do use this approach, you can then also align the images, find the same regions, etc., etc. So now I would like to show you a brief video, if it works. So here, for instance, have the 3D, the reconstruction of the cell. This is a eukaryotic cell. You can clearly see the ultrastructural features, individual mitochondria, the nucleus, the nuclear membrane, the nucleus inside. And once you obtain such a full reconstruction, you can also think about doing statistics on such a sample. Still bearing in mind that the level of information we obtain is limited to the ultrastructure, actually. Okay, let's quickly go further. All right. So then the last part shall focus on the cryo techniques. Since the room temperature techniques we talked about before cannot bring the utilization of the electron microscopy into the region of resolutions where you would like to be in order to prepare high resolution studies, an alternative approach is to quickly freeze your sample. And we have two general approaches for freezing the sample. One is called plunge freezing, which is a rapid immersion of your specimen into the cryogen. The cryogen here is a liquid ethane or ethane propane mixture. The immersion has to be very fast. The sample has to be frozen at a rate of roughly 10 to the power of 4 to 5 kelvins per second. And for the life science types of the samples, these techniques can be only used for the samples which are thicker, less than 10 microns, or which are less than 10 microns in thickness. An alternative approach is the so-called high-pressure freezing, that is the freezing by the liquid nitrogen at the presence of the very high pressure, in general normally 2000 bars, and this will allow you to freeze the water into the vitreous state, this glassy-like water, even when the sample is around 200 microns thick. For plunge freezing, we primarily place a low amount of your sample on the TEM grid. This might not necessarily be only your protein, but it can also be bacterial cells or cells adhered to the TEM grid. You remove most of the liquid by blotting, by filter paper, and then rapidly immerse into the liquid ethane. For plunge high pressure freezing, the sample is placed into this carrier, into the filler, so-called filler. The volume is completely filled, and then you use this micro instrument that creates very high pressure and sprays the liquid nitrogen onto this carrier to freeze it completely. As an alternative approach and combination that improves the issues that have been discussed for the block phase techniques. The high pressure freezing is sometimes coupled, or very often coupled, with the freeze substitution, where the sample is first frozen using high pressure freezer, but then the water is replaced by the mixture of the acetone with the fixative and the contrasting agents at the temperatures which are just below minus 100 degrees Celsius for a certain period of time, and subsequently this the acetone is replaced with the resin, and you can again bake such samples to do the block phase imaging at the room temperature, which brings the advantage of preservation of additional ultrastructure information, which may be lost due to the dehydration when the samples are prepared at a room temperature from the very beginning. Of course, if you have the bulky specimen, which you cannot image in the transmission electron microscope because it's more than half a micron thick, one obvious thing is to cut it into the slices. For that, the technique of so-called semovis has been developed, which is a CryoEM of vitreous section. You'll take the high pressure frozen sample and use the ultramicroton, but operate it at the liquid nitrogen temperature, so your knife is cold, and you float the sample or individual sections on the liquid nitrogen level. If you manage to prepare these thin sections, then the sample contains even a much higher level of detail, because the contrast is now primarily from the protein composition of your specimen, not from the contrasting agents that you used using the sample prep. So you see the natural contrast, which brings the problem that the contrast is low. However, this approach is very close to the native state, because you're now imaging your specimen in such so-called near native state. Still, this approach has certain issues, which are primarily concerned with releasing of the surface tension due to the slicing, and you probably will see, in general, you see different cartons. The surface is not fully flat, which may lead to presence of additional artifacts, specifically in the direction of the cutting. For that reason, there is currently a technique which combines focused ion beam micromachining at the cryo temperatures to prepare thin cellular sections. So you insert your specimen, frozen specimen, into the scanning electron microscope and use the gallium ions to ablate the sample and prepare a thin lamella, which is then transparent for the transmission electron microscope, and you transfer such lamella to the transmission electron microscope for cryo-electron tomography. And the process works like this. For instance, you have a TEM grid with the adhered monolayered cells, which were in this case plunge frozen. You select one of these cells, select the regions which you want to remove, and scan with the gallium ions in these regions, and you end up with a thin section of the cell that is then electron transparent for the TEM, and where you can look at your sample of interest. And here, for instance, these are the results from the group of Pavel Plevka, primarily done by Jana Marovcová. Here you have, then you can study, for instance, the virus life cycle inside the cell. This is the vaccinia virus. Or you can look into the eukaryotic cell. This is a section from the HeLa cells. We can nicely see linear and circular polysomes. And thank you for your attention.